Thank you, sir.
So I can share the slides?
Yes, sir. Sir, there will be a short introduction, then we can, sir.
So you have give me the clue for the share the slides, okay?
Yes, yes. Sam will give you, sir. Yes, sir.
Okay. Welcome back to the lecture workshop on the OMIC sciences in the times of COVID pandemic. We hope you had a hearty meal, and now we will resume with the afternoon sessions. I now hand over the platform to moderator Ritika, student of first year MSc Microbiology.
Thank you, Candida. Good afternoon to one and all present here. I take immense pride to welcome back our guest of honor, Dr. Dipankar Chatterji, an honorary professor of Molecular Biophysics Unit at Indian Institute of Science, Bangalore, and is also a recipient of the Padma Shri, which he acquired in the year 2016. Sir had mesmerized us with his enriching words on the topic antibiotic resistance in his yesterday's lecture. I hope you all are geared up for a second round of enrichment from the topic social behavior of bacteria in today's session. Before which, I would like to inform the participants to type out their queries in the chat box given below, which will be addressed post the talk. Over to you, sir.
Thank you. I have to go to the share mode for the slides. Somebody.
Yes, sir. Share cost and then share it, sir. Sam?
Yes, ma'am, sir has been made a presenter.
Okay.
Sir, to share the slide, we may not know everyone.
So on the bottom of your screen, you should see an option called share.
I don't see that. Yesterday did not come. Share here.
Yes, sir.
Okay.
Yes, sir.
So shall we start?
Yes, sir.
Okay. So yesterday we have been talking about antibiotic resistance. I hope I could convey the message which I want to project, that no matter it is a COVID time, virus infection or bacterial infection, we have a major issue for having different therapeutic possibilities, and antibiotics are the biggest saver of mankind by acting against bacteria. However, with our reckless use of antibiotic, we created problem for ourselves. Though antibiotics directly do not affect virus infection, they do affect the secondary infection. And we set different strategies by which the antibiotics were developed in the market. Most of the antibiotics focus on central dogma pathway, that is DNA synthesis, RNA synthesis, protein synthesis. RNA polymerase and ribosomes are greatest target for these antibiotics. Even DNA replication is also targeted. We shade that different ways DNA structure, compaction of DNA, chromatin formation with acetylation, methylation, epigenetics modification of histones. They are all very wonderful pathways for developing inhibitors. What all want to do is to develop inhibitors which are specific for bacteria or the infection, not affecting the host very much, creating minimum toxicity. During the process, what we encountered over the decades, the bacterial biggest, any living being, I mentioned it repeatedly, I'm mentioning it again, that the existence of the bacteria, survival, is the most important, is the hallmark of the existence, is the struggle and survive. This struggling and surviving gives rise to different mechanistic pathway, and then there are many mutations. The mutations in the RNA synthesis and protein synthesis pathways are very well known, so resistant mutants come. By prolonged use with sublethal concentration of antibiotics also create tremendous problem for antibiotic resistance. So to circumvent that, we thought of deciding on different pathways which are very important for bacterial existence, and those pathways as the stringent response pathway, quorum sensing pathway, or the pathways which are not directly necessary for survival, but the pathways which are keeping the bacteria alive under very difficult conditions. Keeping that in mind, I'll come to the social behavior in bacteria. My today's talk, it will be more like a story and very new observations which have been made in the literature, which are directly linked to the antibiotic resistance, development of the resistance, and working with them. What do you mean by social behavior? The social behavior is defined from Latin word friend or allied, which is basically what means that how do people interact with each other and how they interact with each other and how they live in a group, not as an individual. Nobody can live as an individual. There's a great training we had that over our existence, that every organism had to live in as a social being together, and that is very important for the survival. So we see different animals, monkeys, penguins, elephants, fishes, they are all living together, not alone. If they stray away, go alone, they become insecure and they are susceptible to die. So bacteria is no exception. Social behavior of honeybee, the workers queen and the drone, you must have known it, all this, studied all this. They are also very social beings. They have their own work cut out for them. Each species has different kind of work, and they together live in a society and perform their function. Man is obviously a social being as we developed, and we can see that finally what we come here is today's men, Facebook. But they are coming from prehistoric time as we develop. But in many cases initially, they were not such a big social animal. However, they understood, realized, and learned to live together. And from the cavemen, Neanderthal, everybody has a social behavior which helped us to understand their we understand their function and existence. So this basically we ask the question, these are all big animals. This social behavior is true for the bacteria or viruses also. I cannot say for viruses because they're non-living things. They live as a parasite on the basis of the other thing, host. But bacteria definitely have this social behavior. We never knew this before, but now we know. And they become a wonderful pathways to kill them, wonderful pathways to attack them and to take them into task and create. And now people understand that those pathways are much more important for killing them other than the nucleic acid or protein synthesis, the central dogma of molecular biology. Bacteria, as we know, there are prokaryotes, unicellular, good and bad, division through binary fission. And these are the E. coli bacteria, the classical common bacteria, which divide nicely through binary fission. So they had different good bacteria there in the gut microbe. You know very well, you must have heard the word microbiota. Gut microbiome, the several bacteria keep our life alive, existence, and also are beneficial for survival. But there are bad bacteria all over who are infectious, will kill us, and we need antibiotics to counter them. What is the need for asexual unicellular organism to display social behavior? This is the question people started asking. It was not a very old concept. It was a new concept when everything was social. And every life species, that very nice question which scientists pose to themselves, that if bacteria follow the same pattern of living like any other higher animals, what is the need for a unicellular organism to display social behavior? Do they have the social behavior or not? Very important question. I hope you understand this. Herbert Spencer showed that he was a contemporary of Darwin. He known for his work, Survival of the Fittest, and popularized altruism. That altruism is nothing but sacrificing oneself for the common good of the community. And he was the first to apply the term altruism to single-cell organism. And he said the bacteria are indeed social. You look at the time, 1820 to 1903. So he was the first person he talked about survival of the fittest. And he said the bacteria are indeed social animals, social and living things. And he used the term altruism, made them popular. And he said that altruism, or killing oneself for the common good of the others, is therefore a very important path for the organism to survive. So they are indeed social organisms. If that is the case, then they should be very easy to handle. If we now think of it this way, that okay, I said there are mutations possible in the central dogma, mutations possible at the very essential pathways. You want to kill a bacteria by tampering with the essential pathways, they create mutation. But if I do it cleverly in a different ways, I kill their social living, I'm not killing directly, but what I'm killing their living together, their communication satellite, communication network. So if we do that, will they be able to survive? Will that be mutation? They will take long time to create a mutation for that because the time gap is very long. Will it be beneficial? That is the question we asked. Russian famous evolutionary biologist Kropotkin told us. He was economist, activist, philologist, zoologist, evolutionary theorist, philosopher. We must prepare to learn someday from the students of microscopical pond lives, fact of unconscious mutual support, even from the life of microorganisms. So this is a time in the turn of 20th century, this concept was coming up that we have a lot to learn from the microorganism. If we learn, we can control them, we can kill them. It is the main idea is to remove them from the human life as much as we can. So social behavior of microorganisms was hardly explored. By the end of the last century, the studies of the social behavior of microorganisms bloomed. Several such, like what I mean by last century is the 20th century. Around 1980, 1970s, 1980s, the initial work was started in bacterial social behavior. So it's a very recent subject. I don't know whether microbiology students in the master's class are taught in these areas, but they're a fascinating subject to teach. And because those are the subjects, why it is important, I'm re-emphasizing myself several times, that these are the pathways one has to control to keep infection under control, or the survival of the bacteria in control. So, if behavior is social, how do you define it? If behavior is social if it has consequence for both the actors and the recipient. What is actor and recipient? Actor and recipient are nothing but, I am a social animal, suppose, consider yourself. You are doing something which is going to be good for your neighbor. Then the neighbor is recipient, you are the actor. If it is vice versa, the neighbor does something and you become happy about that, then you are the recipient, neighbor is the actor. So social behavior can be categorized according to the fitness consequences they entail from the actor and recipient. So we mean it basically that a behavior is social if it has consequences for both for the actor and the recipient. And social behaviors can be categorized according to the fitness consequences they entail for the actor and recipient. And these are the effect of the recipient. You look at very carefully. Classification of social behavior. When it is mutual benefit, both actor and recipient both are happy together. Actor eats something, recipient eats something in a function. During Ganesha festival or Diwali, you see your neighbor cook some food, give it to you. You cook some food, give it to your neighbor. Both are happy. It's a mutual benefit for each other. It's a perfect social behavior. Selfishness is that you only receive the food, but you don't give it to the neighbor, and it's bound to die. It's called selfishness. Altruism, that you sacrifice, you don't eat at home, you give it to your neighbor, for whose good your neighbor is good, or for the common good of the society. There are many people sometimes don't eat and give it food for some political cause or some development of children in a very difficult society, impoverished society. These are altruism and spite, both are dangerous to each other. They fight and they don't talk with each other. So these are the effects. So effect on recipient, I classify them as 4 ways: social behavior. These are the effect on actor and these are the effect on recipient. So you have mutual benefit, selfishness, altruism, spite. These social behavior, all of them are exhibited by the bacteria. And secret social behavior, secreted mechanisms, what are the important for social behavior in bacteria? What are the communication network? They have enzymes, extracellular substances, cytophores, autoinducers. These are the different kind of small chemical molecules, mostly small, not very large macromolecule, not like DNA, RNA, protein, not large molecule. If there are protein also, there are small protein molecules. But there are enzymes also involved, which are there, which are creating the social behavior active. So you have extracellular substances, cytophore, autoinducers, etc. So these are iron carrier cytophores. These proteins, these molecules, they carry iron to the place of necessity and do the work. So actor eliminates this kind of molecules to take iron to the recipient if his iron shortage is there and give rise to this kind of behavior which is dependent on each other. Selfishness or cheating, these are called tragedy of commons. It's a very important, very interesting English term, tragedy of commons. Pseudomonas aeruginosa form biofilm in the last of cystic fibrosis patients and secreted cytophores to maintain infection. So Pseudomonas infect and secreted these cytophores. Found that some cells do not secrete cytophore. This reduces the overall virulence. And selfishness is bad in the long run. This is what I wanted to tell you. That in the previous slide we said that these are the molecules. The previous slide we said that there are small molecules which come, help each other, and these molecules are synthesizing and sending this to carry iron to people who cannot have that. You need to have, when a Pseudomonas aeruginosa form biofilm in the last of cystic fibrosis patients. See, the point is here, always remember, they are getting killed by their behavior, so it is bad for them, but it's good for us. So they have to maintain themselves, which is good for them. So they like to produce several cytophores, secret cytophores to maintain infection. They would like to maintain the infection on cystic fibrosis patients by biofilm-dependent infection, and we won't like to have that. The some cells do not secrete cytophores. This reduces the overall virulence. And selfishness is bad in the long run. So if some cells want to consume the iron themselves, Pseudomonas aeruginosa, don't give it to their neighbors. They will die. This is a great learning from the social behavior. You understand that if your neighbor is not eating properly, eventually that will affect you. Exactly like COVID situation now, if a social animal, we are all social animals. If your neighbor is COVID infected, doesn't move around with a mask, doesn't tell anybody that he is or she is a COVID infected, eventually it is bad for them also. So selfishness is eventually bad for social living. And that is a great teaching for social behavior. And those can be attacked for our good, for the recipient's good. Tragedy of commons. So there is a public good, there is a cooperativity, there is a checker cells, and this goes from one cell to the other, and they live together very happily ever after. Mutual benefit. What is the mutual benefit? That you have produced something and you infect something, you go and make a residence in some location, and you some bug you use for your survival. So it benefits to each other and they live together very happily. So it's a symbiotic relationship between the actor and the recipient. So any symbiotic relationship between actor and recipient is good for the common good. So if you impair the pathway for the symbiotic relationship, you can definitely do the job very well. Altruism, these are toxin antitoxin magic. I am not going to explain antitoxin toxin system very much. These are very well-known thing. That toxin antitoxin system of Escherichia coli. You infect a toxin which have antitoxin coming along with it, and they together maintain the infection. So altruism, that when it's under survival become questionable. Spread of antibiotic resistance, this is one of the problems. Now, if I use antibiotics to kill infection, sensitive population with very few resistant mutant. But as we go on, yesterday I gave you an example how to grow the infection, how to grow the antibiotic resistance mutant. That if you go on serial dilution, you will get a tremendous number of colonies coming, increased number of colonies coming slowly. So you come with a low concentration of the antibiotics. You generate few mutants. You take those mutants and plate it, use it for the growth in higher concentration of antibiotics. So as you go on increasing the higher concentration of antibiotics, you can reach a higher, very high concentration. So enrichment, considering sensitivity to population, you have it only one, but it goes up if you culture it properly in the presence of antibiotic pressure, and entire population become resistant. So this is the most dangerous thing. So you have to have a pressure all the time in the presence of antibiotics. Organism is trying to survive. Organism trying to manipulate itself to survive properly. So I think I mentioned it yesterday also. Antibiotic resistance is a phenotype acquired by bacteria during prolonged treatment due to selection pressure, attributed to by several factors like mutations in the antibiotic binding site or due to other physiological changes. So how to counter that? This I showed yesterday. I'm repeating myself that antibiotic development, antibiotic resistant observations always see something comes in 1930, within 10 years we get a mutation. Something came penicillin in 1945, and a mutation came in 45, by 40 there, in discovery of the antibiotic. So like that, every single antibiotic, I explained the slide yesterday, every single antibiotic has a deployment and the resistance development go hand in hand, parallel to each other. This I also showed you. These antibiotic resistance not come directly by us, but also from the animal and the feed. Food we eat, because the feed is also antibiotic resistant because of the profound use of antibiotic. So these are very common. Again, I emphasize to all the people who are listening to me, in post-antibiotic era in which common infection and minor injuries can kill. Nowadays, if I have a small cut, I put Dettol, I get survive, but it may not happen like that if we have resistance development. So be careful. Far-forming apocalyptic fantasy instead a very real possibility for the 21st century. It's a very important writing. Keep it in mind. So altruistic defense, this is called altruistic defense, is that sublethal concentration of antibiotics, slow growth of sublethal antibiotic, and you slow growth further and you make it total population antibiotic resistant. This I have taken from 2010 paper. The process repeated 10 times with increasing concentration of antibiotics. Most of the cells became antibiotic sensitive, only very few cells were antibiotic resistant. These highly resistant isolate produce the signaling molecules indole, which activates drug efflux pump, other protective mechanism is susceptible kin. These altruism allows weaker constituents to survive and to have the chance of beneficial mutation. So these are all chemical. There are different efflux pump molecules are produced which tell us to go for antibiotic resistance. In the case of spite, bacteriocins and antimicrobial toxins produced by bacteria that target closely related strains and species and to which the strains is immune. And this goes on in what happened that in both the bacteria happened to infect and host caterpillar, the mortality caterpillar is reduced. So spite is a phenomena where there is a mutual antagonism between the two infecting organisms and they survive accordingly. So these are the experimental proof for the spite. This has been shown in great detail in this particular paper that mortality they assay as a function of co-infection and they find one helps the other or one kills the other depending upon the condition. This is a very important thing. We never knew this exists, but now it has come in 2003 science paper. They showed that bacteria show cannibalistic behavior in Bacillus subtilis, a gram-positive organism. That if you are inactive, you can have a cannibalistic behavior. Sporulation of a killing factor, sporulation of a delay factor. So these are is an energy incentive process that is irreversible after the early stages. If food resource becomes available during later stage sporulation, cell would be a disadvantage related to bacteria that could initiate growth immediately. For the bacterial population as a whole, it is beneficial to delay the onset of sporulation. So sporulation onset, they have to delay. There is a simple biology behind it. And this can happen by cannibalistic behavior in bacillus. But they also have altruistic behavior. They kill each other to make the other people survive. Why they do the altruistic behavior? If there is a population of bacteria, they are under starvation. They realize from their internal signaling, they realize that the condition is very bad and some are more sick than the other. So the sicker population, or in a way older population, they kill themselves so that after their death, their metabolic products can be utilized by the young living ones so that the survival can continue, or the species can continue. So maintain the species, altruistic behavior comes, but when it is not so dangerous, then they kill each other to survive themselves. So it is a cannibalistic behavior also. So in the roots of the plant, this kind of social behavior is very well known, rhizobiome. The studies have shown that there are up to 10 billion bacterial cells per gram of soil in and around the plant root, a region known as rhizosphere. And dozens of species appear to suppress plant disease by excreting substances that ward off pathogenic microbes or occupy niches otherwise taken up by the pathogens. So rhizome depends heavily on the soil type. So these are different ways of survival of the organism. Nitrogen fixation by legumes is a classical example of this kind of survival, and that goes by several chemical pathways. Biological nitrogen fixation is a process that changes the inert nitrogen into biological use for ammonia. So these are various examples I'm showing you. All the different behavior of social behavior bacteria shows how they can be manipulated, how they can be seen in different species organisms, which are now accumulated last 20-30 years to understand the social behavior in bacteria. And the major question is that by impairing the social behavior pathways, the mechanism of social behavior, can we play around with the survival of the bacterial species? Now, this is a very important thing which has just come last 20 years. Gut microbiome. It helps the body to digest certain food that stomach and small intestine have not been able to digest. So our gut is full of bacteria. In fact, so much so, very recent finding, I don't have the slide for that, last 2 years, it has been shown that all our behavior, our mood, our anxiety, our mental faculties, all depends upon the food we eat and our microbiome. So a person A and person B have different kind of gut microbiome. It has been shown that people living in very developed society, in a developed place, their food items habits are so different, their microbiome will be different, but not so much different from the people who are coming from poverty-stricken places. But if you have more food and a very healthy food, your microbiome profile will be different. What is gut microbiome? Number of bacteria, different kind of species of bacteria which are present in your gut, and they manifest your behavior, your anxiety, your health, your mental faculties. These are very well documented now. The connection between neuronal behavior and also your gut microbiota. In the immune system, performing a barrier effect, healthy and balanced gut microbiome is key to ensure proper digestive function. It helps us combat aggression by other microorganisms, mainly the whole cell of the intestinal mucosa. So therefore, this is also another very important area. Can we ever kill this gut microbiome? We cannot, and we should not, because many of them are very helpful to us. They help our survival, help our habit, our livelihood, our immunity. So much so, we should not kill them. We have to manipulate that so that the bad ones we can control. Competition for resources is a very important, it's called restaurant hypothesis. This is also from Microbes in 2015 August, American Society for Microbiology Journal. This invading pathogen must compete with the resistance microbiota to enter a restaurant or they will be eliminated. This is called restaurant hypothesis, that if all the food are eaten by the guest in a restaurant, no new person can come in. So invading pathogens must compete with the resident microbiota to enter into a restaurant or they will be eliminated. So there is a fight going on for the access, for the food, for the survival. And these are also pathways to kill the bacteria. So social behavior becomes a very important part of the action. That is the whole point I'm trying to emphasize in this talk. Food for thought. This is very important. Again, is the 2013 data. Number of human cells in 10,013 and number of human cells is 10,013. Number of bacteria on humans 10,014. Isn't it amazing? Number of human genes is 30,000. Number of bacterial genes in human is so much more than 30,000, right? So therefore people think this is a cover picture when it was published. It is a sensation in the world. So it becomes a cover picture in this very famous Nature Scientific Journal. And people think that we are bacteria basically. We are nothing, we are not human beings, we are bacteria. We are just having living, bacteria is together utilizing us to grow and survive. We are just part of bacterial population because the amount of DNA we have, amount of genes we have, much less the number of bacterial genomes. So gut microbiota therefore becomes very interesting. It's so difficult to study all of them. Which is useful, which is not useful, becomes a major important point. How they talk with each other, I made it a point yesterday to tell you about quorum sensing by today. I'm talking about that now. Microbes also display impressive ability to detect the density of their own and other species through quorum sensing. Quorum sensing is nothing but communication between bacteria network, how they talk with each other to make them survive under different conditions. And under different condition. And is a gathering, quorum is a beautiful word taken from the English dictionary. A gathering of the minimum number of members or organization to conduct business. Quorum sensing is sensitive who is around, sensing who is around. So if there is enemy coming, one will tell the other, be careful, enemy is approaching. Involves a wide variety of secreted compound known as autoinducers, some pathogen vesicles. If cells secrete a chemical, then as cell density goes up, so will the local concentration of the chemical. By responding to the chemical, therefore, the cells respond to the density. Quorum sensing actually tells us that if we can address these quorum sensing pathways, if we can kill the communication between bacteria, if we can kill the quorum sensing, if we can stop the autoinducers, we'll be able to handle them. We'll be able to kill the bacteria. Because remember, I'm a smart student will immediately get the point that in a social behavior we are attacking, what we are attacking basically the social living of the bacteria. In a gathering of hundreds of bacteria, suppose hypothetical number, if I can kill the communication, then if 5 becomes separated from 100, those 5 will die faster than the rest of the 95 because they have become antisocial. So that becomes a very important point to treat. Quorum sensing is ultimately leads to biofilm formation. Some question came yesterday for me after my talk yesterday, how antibiotic can penetrate biofilm. I was waiting for this slide. Biofilm formation is guided by the quorum sensing, and biofilm formation by non-motile bacteria, they form a biofilm of this organism. They also form by motile bacteria with flagellar movement. And these biofilms which are produced over the surface, any plastic surface, synthetic surface, cellular surface, anywhere, those catheter, implants, heart pacemakers, they're dangerous. You cannot kill the bacteria. They infect you and antibiotic cannot pass through the biofilms. These are coming because of the social behavior. They're trying to survive under onslaught of antibiotics. These are different autoinducers which create oligopeptide acyl homoserine lactones, those who are responsible for the quorum sensing. A city of microbes is a structured compound of bacteria embedded in a cell-producing polymeric matrix and attached to abiotic or living surface. Can be composed of multiple species or singles. Biofilm protect bacteria from antibiotics and other environmental insults. They are caused of nosocomial infection as well as persistent infection. I have worked on long back. I worked on bacterial biofilm, and this is one of the paper I published, which is taken as a cover picture of the journal Microbiology. These are nothing but the bacteria, biofilm, Microbacterium spigmatis biofilm, MCT125155, grown under a water interface in a plastic dish. We routinely grow them, and you can see it's like a cream in a boiled milk. Cream forms on the surface of the milk vessel is basically similar kind of texture, layers of them, and it's very difficult to penetrate them. Altruism in bacteria, Microbacterium spigmatis biofilm. We have seen this biofilm, this is also coming from my own laboratory, that they form a beautiful biofilm. I showed you in the previous slide how the biofilm is formed. Here I go into the deeper in the microscopy and show a city of microbes. They form a network, close network among each other. And to kill them, you have to penetrate them, which is a tall task. You can see the dental biofilm. It's not a very pleasant picture to see. This is a Microbacterium spigmatis biofilm. This is a dental biofilm, multi-species biofilm image taken from a Google image that you see if you don't brush it properly or your mouth, you get oral infection, you get such biofilm over your teeth, which cannot go after some time by the brushing also. So removal of the teeth or the neighboring teeth are becomes important parameters. Biologically, there are many stories. One of the stories which became a very popular by Richard Lossig group showed that the D amino acids comprising of D-leucine, D-methane, D-tyrosine, D-tryptophan, and very low concentration trigger biofilm disassembly. So they become a very nice antibiotic, very nice drug, not antibiotic, to kill the biofilms because they go for biofilm disassembly. So keeping that thing in mind, we ask the question, how to handle biofilm is basically by handling their disassembly or their maturation. So there should be compounds which are not helping their maturation or helping them disassembly. Same Richard Lossig group reported that the amino acids indirectly inhibit biofilm formation by interfering with protein synthesis. This is the molecule we talked about yesterday. Cyclic Gmp is a beautiful molecule. Cyclic Gmp, C-Gmp, which has a guanosine molecule and a guanosine molecule connected with a phosphodiester linkage to 3' for 5' phosphodiester bond. One through this way, one to 5', this is to the 5', and this to the 3'. They together form a network with a beautiful symmetric structure, which form a cyclic cyclase. So formation of biofilm takes place. These are the molecules. So we thought of mimicking cyclic Gmp, the last structure which I showed you. If we mimic this structure, that would be a very big inhibitor of biofilm formation. In fact, we indeed showed it. This is a published work. And as I told you yesterday, that vitamin C can be inhibitor for rail because of the structural homology. Or remdesivir is a nucleotide analog which stops RNA synthesis. Like that, if I have a modification of the structure, I mimic this site, and therefore I become inhibitor for the formation of this molecule and biofilm formation, and therefore survival of the bacteria. So these are very logical thought goes on after another, make it a wonderful proposition. So the monolayer of bacteria from biofilm, then from we can have a microscopy, microcolony formation. The molecules come together, they attach to a surface, they form a microcolony, they bond together, and then they mature and they form the biofilm. So we try to kill it either at this stage or this stage. Addition you stop, or I stop the biofilm development and maturation. Then you can kill the biofilm. Go ahead. Persistence is therefore a very important parameter. This is the point I was telling you. The persistence of the bacteria. Yesterday I mentioned this. I did not draw the structure here. That if I have a large amount of bacteria growing here, if I use antibiotic, it comes down, but with time, but it will never touch zero. I mentioned it yesterday. So these become persisters. There is a subpopulation, 1%, 2%, which is surviving because of their mutation or whatever the condition they are. They are the infected. They infect organism. I talked about it. So the killing curve is biphasic. Rapid decay and slow decay, and then parallel to the x-axis. Our job is to connect it with the x-axis, make it 100% zero. And that's the whole point. So we go for next slide, where we show that stress and starvation are very important parameters, are very important areas for the formation of several molecules which give rise to survival mechanism, antibiotic tolerance, persistence, toxin anti-toxin modulate activation. So if we treat with the antibiotics, we want to stop this stress molecule. We make analog of these molecules, guanosine tetra pentaphosphate as I showed, or cyclic Gmp, which are forming helping formation of biofilms. We can indeed play around with the survival of the persistent cells. Stringent control I talked in detail yesterday, so I will not touch it upon very much. These are the stringent control gives rise to. Hello, I'll call you little later. I'm in webinar. So this shows RNA polymerase stringent response, and these molecules are synthesized magic spots, PPPG. These analogs are becoming a very important area for development. So stringent response in mycobacteria are very well known work, very well known pathways to develop inhibitors. So I focus on two molecules, cyclic Gmp and PPGP, whose analogs will be wonderful drugs to kill the starvation response, stress response, persistence, growth. We have not proven it yet, but we have shown with some publication that indeed we can create such kind of molecules which will inhibit the formation of guanosine tetra pentaphosphate or cyclic Gmp. Cyclic Gmp was known. These three are called second messengers, and you see the chronology. It started in 1957, then 1969, then 1974, and then 1995 cyclic Gmp. So all these guanosine derivatives are nucleotide derivatives which comes together and play a very important role for the survival. This is the molecule I mentioned before. It was discovered 1987, several years back. This is the molecule. And this is the paper which was discovered 25 years in universal bacteria second messages. So people think, therefore, if we attack this molecule, we make a mimic of this molecule, they'll become wonderful drug or guanosine tetra pentaphosphate. So this is the time lag from 57 to 2012, how the molecules have developed. It started with cyclic GMP, PPPGP, cyclic GMP, cyclic DiGMP. I worked up to this. This is now my current students are working in my lab on cyclic DiMP. It's a completely new second messengers. Its mode of action is entirely different and very, very fine molecule. What is the message? That we have all these second messengers, AMP, GMP. What is the function? Their function is to keep the bacteria alive, somehow under stress. So if we make a mimic, if we make an inhibitor for them, then bacterial survival will be questioned. So I stop my talk here. So with this idea in my mind, therefore, the different inhibitor design will be a wonderful pathway for the second messengers to stop the biofilm, stop the infection, stop the social behavior, and therefore becomes a beautiful pathway to run further. Thank you very much for your patience. I'll take questions.
Thank you, sir, for that very enriching talk. We will now move on to the Q&A session. The first question is to treat the biofilm, it's important to know the whole genome sequence.
Not exactly, because we know the partial biofilm information. To treat the biofilm, it helps if you know the whole genome sequence, you can develop inhibitors for that. But without knowing that also, If you know what are the small molecules which are produced, autoinducers, second messengers, autoinducers, acyl homocysteine lactone-like molecules, you can target them for the inhibition of biofilm formation.
Thank you, sir. The next question is, can bacterial pathogens be confused by using peptides as quorum sensing? Asks Arun Kumar.
Could not follow the question. Repeat again.
Can bacterial pathogens be used? Sorry. Can bacterial pathogen be confused by using peptides as quorum sensing?
Yes, they can be confused, but what is important here, you are using the sublethal concentration of the peptides, which are again autoinducers, and therefore it is acting as a autoinducer for the cellular development or the communication between network. So the confusion will not come because that come in the much later stage. But it will, basic aim is to stop the pathway for formation of the biofilm, and therefore our quorum sensing pathway. So it doesn't really matter what finally pathogen does by looking at the peptides. It will not reach that concentration yet. Yeah.
Oh, okay. The next question is asked by Dr. Poppy Dutta. Does all organisms, viruses, fungi, etc., show social behavior?
See, I was very careful in mentioning that bacteria do. Fungi do, but I am not so sure about virus because they are not living species, they are parasites. They live only on the host. Upon infection of the virus, see, I'm sure you know about it, that when virus infects, many viruses stay in the prophase stage, right? They integrate with the chromosome, in the host chromosome, and they lie low, they are absolutely non-infectious, they finally goes on multiplying with the chromosome. But their behavior change in induction, they become lytic, classical lambda repressor model. So prophase to lytic or lysogenic to lytic phase modulation is a behavioral change. Whether that is close to telling that they are showing under different condition, is like a social behavior or not, I cannot tell that, but appears to be so. But no direct proof yet social behavior directly, like all the parameters of social behavior, like spite, altruism, cannibalism, recipient, actor, all that not so well defined in virus population. Subject of research has to be done. But lysogenic to lytic conversion is a very important social behavior also.
Okay, thank you, sir. The next question is by Ms. Sharmili Kamal. Are there any known natural quorum quenching substances?
Good question. I cannot tell you right away, but people predict that certain molecules or metabolic food products which we eat, they can act on bacteria who are long survival for a long time. But I don't think there are major proof of that yet. Some food products. Not much science, and I'm not aware of it.
Thank you, sir. The next question is by Dr. Beatrice. What if two organisms having biofilm put together, what changes could be expected?
What are the two organisms?
So the question is if two organisms having biofilm.
Yes. I don't know what will happen. I don't remember any. I can't tell you offhand whether any experiment has been done that I have a mixed population of bacterial culture, both from, I don't think any problem, both will from biofilm. But whether one inhibit the other or not, I don't know. One can study that.
Thank you, sir. The next question is again by Ms. Sharmili Kamal. I've heard of fratricide mechanism that is seen in biofilms where cells kill their kin for building ECM. Can you throw some more light on this phenomenon?
Chain of what? Chain of?
Fratricide mechanism that is seen in biofilms where cells kill their kin for building extracellular matrix.
Yes,
Can you throw
yes,
some?
yes. It is there. I mentioned it in my presentation in some slides that in rhizobium or some places it has been shown that one can affect the other cell to making that to help each other or sometimes to stop it. Most of the time they try to stop the others to do that so that they can perform function. It's a typical selfish behavior, which is not good for them. So social behavior they cannot survive very long, but there is demonstrated. They're demonstrated.
Oh, thank you, sir. That was the last question for this session.
Thank you very much.
Oh no, sir, 1 minute. We have another question. Silver nanoparticles of various plant extracts have been found to target AHL molecules.
Yes.
Have these
yes.
inhibitors
Yeah, absolutely. I don't know. It's a very knowledgeable
that could
question.
prevent the formation of biofilms?
Very good question. It's a very knowledgeable question. Acyl homocysteine lactone formation is inhibited by silver nanoparticles. This has been demonstrated, and therefore it can be utilized very efficiently for stopping the biofilm formation. People have tried this. Whether it can be used as a medicine or not, I do not know yet. But people tried this.
Okay, sir. Thank you, sir, for patiently answering the questions.
Thank you very much.
Thank you, participants, for your ardent involvement in this session. The questions surely made us well acquainted with the topic.
So I can go from this, right?
Yes, sir, you can.
Thank you so much, sir.
Thank you very much.
Thank you very much.
Participants, please note, the second session will begin as per the schedule at 2:30 p.m. I request all the participants to stay logged in and be back at 2:25 p.m. for the second session. Thank you all for joining us.